You, so just interrupt me. Okay, hello, I'm Helen Willsey. I have a lab at UCSF where we study many different genes. One of the ones we've been studying the most intensely has been DYRK1A, and the work I'm going to show you today came directly from one of these meetings a couple years ago and some conversations I had with parents. Please feel free to reach out to me. This work was just put on bioRxiv. The QR code is here. It's by graduate student in the lab, Kate McCluskey. It's under review right now at a journal, and I'll show you this QR code again at the end if you want it. So the real story I'm going to tell you today about DYRK1A and gut neuron development, but let me give you a little background first. So my lab studies neurodevelopmental disorders and the genetics of neurodevelopmental disorders. Primarily, we study autism, and so these are some of the genes that carry the most risk individually for autism, and DYRK1A is on this list. So that's how we started getting interested in DYRK1A, but we have now developed a much deeper interest in it than just with its association with autism. So because we study over 100 genes at any given moment, we use a scalable genetic model, which are diploid frogs, and specifically Xenobus tropicalis. Unlike zebrafish, for example, frogs have a simple diploid genome with one copy for mom, one copy for dad, so it makes the genetics far more straightforward. One mating pair can lay over 4,000 embryos in a day. So that dish under there is actually a 15-centimeter dish, so very large. So we have a ton of embryos to work with. They develop extremely rapidly. In just 6 or 7 days, you have an eating, feeding, sleeping, behaving tadpole that you can do a lot of different assays on. They're very cheap compared to rodent models. And the drug screening is very straightforward because you just put the drugs in the water, and genetic manipulation is also straightforward. And also I think there's this key idea of the DYRK1A protein is highly conserved between human and frog. So here is just the amino acid sequence of the protein itself, comparing on the top line the human version of DYRK1A and on the bottom line the frog version. And just showing every letter here that is red is the identical amino acid in the protein between the human and the frog. So it's a highly conserved protein. And so we feel like the findings that we'll have in the frog will likely translate, at least generally, to humans. The coolest thing about frogs, and if you only remember one thing about frogs, let it be this, is that we can take embryos and catch them right after that first cell division of life at the 2-cell stage, and we can inject CRISPR-Cas9 reagents that can mutate any gene we want, along with a red dye. And we can create a tadpole where, for example, just the right half of the tadpole carries our mutation of interest. And then we can compare what happens during development on that side compared to the other side of the animal. So we have an internal control within our animal to compare to start to understand and elaborate gene function. It's fast, 6 days, $25 per gene. So again, this is a scalable model organism to study many genes in parallel. So our typical afternoon where we're making hundreds of half mutant animals. This is just a proof of principle where on the right side of this tadpole, I've mutated the gene required for eye pigmentation. And so you can see on the right side, it has a white eye, and the left side, which is the control, has a pigmented eye. And so now we can infer that the function of this gene is an eye pigmentation. The nervous system of the tadpole is exceedingly beautiful. This is an antibody stain just to look at all of its neurons. But we don't have to just do that. We can look at particular subpopulations of neurons, progenitor neurons, excitatory, inhibitory, astrocytes, microglia. So in one animal, we can have all of the typical cell types present generally, and be able to, within the animal, compare what happens when you mutate a gene. I've also done a lot of innovation in terms of imaging modalities in the system. Because of the size, we can image the whole thing in 3D and do volumetric measurements, cell counts, comparing our injected side to our uninjected side, looking at even ventral structures like the hypothalamus, et cetera, et cetera. Okay, so I'm just briefly going to talk about what we've done in DYRK1A that's already published, and just encourage you, if you're interested in learning more, to go to these 2 publications from 2020 and 2021. But to summarize those papers, what we found was that when we do this half and half model where we mutate DYRK1A on just one half of the embryo, what we see is a reduction in the size of the forebrain. So that's what that red arrow is pointing to. That lobe at the top is the forebrain of the frog tadpole. And you can see how much smaller it is than the other half of the animal that's been unmanipulated. And we went deeper into sort of why that happens, and we did drug screening to try to reverse it. And one of the themes that came out of that was this essential role of DYRK1A in the formation of a cell structure called cilia. So what I'm showing you here on the left with the control is a ciliated cell, and all those pink hair-like structures that are coming out of that one cell are called cilia. And in the respiratory system, and I'll get more into what cilia are, those are actually beating to move mucus in the respiratory system and in the ventricles of the brain. And what we found was that when we mutated DYRK1A, there were defects in the formation of those cilia. And so there's kind of two flavors of cilia in the body, but what I really want to emphasize is that every cell in the body, other than red blood cells, which are weird to begin with because they don't even have a nucleus, but every cell pretty much in the body has a cilia, including neurons. So on the left here, I'm showing you motile cilia. These are the kind of cilia that are in the respiratory airways and the ventricles of the brain and sperm. And they're actually physically beating. In the lungs, they're beating to move mucus and provide a barrier to infection. But then in all other cell types, there's a singular non-motile cilium that comes out from the cell, usually near the nucleus. And so this is more like what a neuron looks like. And this cell is actually a human neural progenitor cell in culture. And that small little pink-like antenna is the cilium. And it may be small, but it is very mighty. And so most of a signaling pathway called hedgehog signaling, which is really important for the development of many organ systems, has to have a proper cilium in order to do signaling through hedgehog signaling. And so hedgehog is really important for cell division. It's really important for developmental patterning of tissues, as well as homeostasis. So defects in cilia can be a big problem. And these are some of the common patient presentations when someone has cilia defects. And so a lot of these will be familiar to your patient group. In particular, epilepsy, heart defects, all sorts of things. So it's a multi-organ spectrum of conditions that can result from defects in cilia. And so DYRK1A having a key role in the formation of cilia points that some of the spectrum of conditions seen in the population may be due to its function at the cilium. And again, because it affects so many different organ systems, this is where the frog can be really nice, because we don't just have to study neurons in a dish. We can study all of these different organ systems and start to understand the function of DYRK1A, not just in the brain, but also in these other tissues that could be of interest. Some of this work has been published, and so we had a really great collaboration with Rachel Miller's lab, who really led this work in frogs, but showing that DYRK1A is highly expressed in the developing kidney in the frog, and that loss of function of DYRK1A caused kidney defects, both in the frogs, and then confirmed that in a patient cohort of 19 folks with DYRK1A pathogenic variants. And so then I was thinking about where to take the DYRK1A project next in the lab, and I went to the DYRK1A family meeting and gave a brief talk, and had some feedback from parents telling me that one of the symptoms that affects quality of life the most is gastrointestinal issues, and has just historically been understudied. So what I'm going to show you today is really our attempt at thinking about why these gastrointestinal issues occur in patients with DYRK1A variants. So like I showed you, I don't just study one gene, I'm usually studying 100. So what I'm showing you here, we took what we learned from the DYRK1A parents and expanded it out to the list of autism genes that we have. But I'll show you here highlighting DYRK1A on that list. And we looked at Simon's Searchlight caregiver survey data. So that's been really helpful. And so thank you to everybody who had participated in the Simon Searchlight, it's being put to use. And so what we see there is in the DYRK1A cohort, 20 out of the 34 affected individuals reported GI symptoms, while sibling or family unaffected members, none of them reported any. And what I'll show you next is the citizen patient medical record data. We don't have it for DYRK1A, but this is my plug to ask you all to think about getting together with Citizen, and I can hook you up with them, because they actually have detailed medical records from each one of these patient cohorts. And so then you can get a lot more granularity about the specific kind of gut symptoms are actually reported. While Simon Searchlight is very broad, sort of GI in general, Citizen, then I can actually get de-identified information about all of the diagnoses that a person has ever had in their medical record. And so you can see for some of these other genes where that data is available, you can see a very high percentage of them show GI dysmotility. So actually movement of the gut, which usually you can see at the bottom is usually constipation. There's certainly folks with diarrhea and vomiting and reflux, but constipation seems to be the biggest contributor to this gut dysmotility. And I think through some conversations with the DYRK1A community, it seems like movement of the bowels seems to be a common issue among the population. So this project has now become a whole part of our lab, led by a graduate student, Kate McCluskey, who is about to graduate. And it led to this, this question of why do autism or the gene variants co-occur with gastrointestinal issues? And again, what I showed you before was that it's really a lot of constipation or inability to move the bowels. And so the movement of the bowels is largely controlled by gut neurons, which are also called enteric neurons. And so they innervate the gut and actually cause motor neurons to then contract and move along the gut to actually move contents through the gut. So we got interested in these gut neurons and understanding their role there. And so when we look at that whole population of autism genes that DYRK1A this applies to, is we see enrichment in expression in human enteric neurons, those gut neurons, as well as their embryonic progenitor cells. And so it seems to be there's something about these populations of those neurons that are in the gut, and then also the cells that they came from earlier in development. And so what we really wanted to ask was, how do mutations in these genes, and especially DYRK1A, affect gut neuron development? And so this is where I really think frogs come to the rescue. Because again, there's a lot of advantages we could do here, right? So I already told you about the diploid genome, the CRISPR editing on one side versus the other, and the number of embryos. But what's really cool is that we can actually watch these neurons migrate into the gut. So they actually start pretty up high in the animal and migrate along, and then start migrating into the gut during development. So we can watch that live in frog embryos. And then you can also see these little dots here that will fire. These are gut neurons expressing GCAP. And so we can actually see the peristalsis happening in the gut. And at the same time, we can see the neurons start to fire. There you go. There you go. You see the gut, then they start to fire once you get peristalsis in the gut. So we can watch these processes live in intact embryos. And then we've now developed assays to be able to watch them, the contents of the gut move. And so we can actually, I can go back, we can actually feed tadpoles fluorescent beads, and we can watch the beads go into the gut, and then we can watch them actually transit through the gut and actually be excreted by the tadpole. And then finally, it's also a nice system because the gut structure is more like humans. While zebrafish just have a tube, frogs have a coiled, complex gut system more similar to humans. And so one of the first things Kate did was she characterized the movement of those progenitors into the gut over time. So she labels these cells with a in situ marker POX2B, and that really labels these migrating progenitor cells that then innervate the gut and are responsible for the peristalsis that happens in gut movement. And so she looked over time, and what you can see is that while they start closer to the head, as the embryo develops, you start to see them now move more into the gut region over time. So we thought that this would be a nice system to now inhibit the genes of interest that we have and see if this process is effective. Oh, you can see them move. Great. And so what she did then was take those two-cell embryo stage, inject CRISPR and a guide RNA against a gene of interest, including DYRK1A. And then you have this nice feature of frogs where one side of the animal has been manipulated and the other side is unmanipulated. And these neurons at these stages don't cross over the midline. So we can actually compare within an animal what happens when you lose that gene to this population of enteric neuron progenitors. And so I'm highlighting DYRK1A here, it's at the bottom right, but we did a number of genes. The top left has the control against that pigmentation gene, and that shouldn't affect this process. And indeed, that CRISPR does not affect this process. The enteric neuron progenitors still innervate the gut. So you can see that red part is totally full. And within the black part. So they've moved into the gut, whereas in contrast, you can look at DYRK1A, where now that red part is a much smaller area and it's more closer to the head, and those neurons have not been able to migrate as far into the gut. Oops. So we went a little further into this specifically for DYRK1A because we have a lot of validated reagents for it from our other work. And so the top is showing you what I already just showed you, is the control and then the DYRK1A, and you can see that in the DYRK1A condition, the neurons do not migrate into the gut as much as in the control. The bottom left is a morpholino, which is just a different way to inhibit DYRK1A through a translation blocking of the protein. We see the same thing. And then we also tried a chemical inhibitor of DYRK1A, TG003, and that also recapitulates the phenotype compared to a DMSO-treated embryos. So we feel confident that the effect that we're seeing is due to DYRK1A loss of function in 3 different ways: CRISPR, morpholino, and pharmacological. Okay, right. So these cells that then become the enteric neurons, in their previous and younger embryos, those are actually called neural crest cells. And so those are a particular kind of cell population born in the early embryo that migrate to a number of different places. So those are the cells that end up becoming the facial bones and are responsible for facial features. But then also, they also populate the peripheral nervous system and especially the gut neurons. So we wanted to look a bit earlier than this and say, well, are there earlier problems in the progenitors of even these cells when you don't have DYRK1A? And so this is that early embryo, and now we're looking down on it. So your left side is the control, and the right side has been manipulated. The far left embryo is a control morpholino, but then you can look next to it, that second embryo. Now we have knocked down DYRK1A on just the right half of the embryo, and you can see a smaller population marked by SOX10. Those are your migratory neural crest cells that end up becoming the facial bones, and then also gut neurons and other things. That third embryo is a control pigmentation gene. You can see both sides look about the same. But then that fourth embryo, now when we use CRISPR to target DYRK1A, that gets reduced. And so we think DYRK1A has this earlier role in neural crest development, and that then also can contribute to the effect on those migratory neurons. This has also been corroborated by a recent preprint from Amanda Dickinson's lab. And so there seems to be now emerging this idea that DYRK1A is really important for early neural crest development, which will affect facial features as well as the other tissues that the neural crest contributes to. And so because we're in frogs, what we wanted to do was try and tease apart these 2 roles. So there's this early role in the neural crest, and then there's this later role in the migration of the neurons into the gut. And so we wanted to try and separate those 2 and ask the question, well, is that migration phenotype just due to these earlier effects in the neural crest? So because we're in frogs, we can leverage their power. And what we did was we let embryos develop neural crests normally. We did nothing to them. They totally, this whole part, that stage of the embryo, we didn't do anything to them. The neural crest developed normally. And then right when that migration starts to go in, that's when we added a chemical inhibitor of DYRK1A. And then again, we asked what happens to migration. So the crest was totally fine. We let it develop normally. And then we specifically inhibited DYRK1A during that window of migration. And so these are the controls. So on the left is a DMSO that should do nothing, and you see the extent of neuron migration in that red region. Below it is another drug that inhibits the RET gene, and this commonly causes Hirschsprung's syndrome, which is so severe that kids have to get their bowels resected or it's lethal. So that's our positive control. That's known to be a problem of neuron migration, and so that looks good compared to the controls. And what does DYRK1A do? So here we're showing you Harmine, though in the graph you also see that other one we showed you, TG003, and what you can see is when we inhibit DYRK1A just during that period of migration of those neurons, again, you still have neural migration problems, even though we let the crest develop normally. And the bottom right is just another control. Sometimes Harmine can also hit monoamine oxidase. So the bottom right is a monoamine oxidase inhibitor, just to show that the effect of Harmine is not due to that. We think it's specific to DYRK1A. You can see on the graph, the far right is that other DYRK1A inhibitor, TG003, that also shows this issue with migration independent of that earlier role in neural crest development. Okay, this is when Kate got real creative. And so she came up with this gut motility assay. So what she does is she takes tadpoles and she feeds them with fluorescent beads for 2 hours. And then she places them, we call them porta potties, and in these little wells that have a 3D printed basket where the poop can go through the basket, but the tadpoles will stay in the basket. And so she lets them defecate for 3 hours, and then she removes the basket. And then she's left with a well plate that has fluorescent beads left behind in the feces from these animals. And now we can use microscopy to count the number of beads and quantify if there's any difference in excretion between groups. And so she did this with the DYRK1A inhibitor, TG003. And indeed, what she saw was that the DYRK1A inhibited group passed fewer beads through their gut. They were still eating. There's other work where she has shown that they actually do fill their gut up. So it's not an eating problem. It really is an excretion issue. So she did a larger screen on unmanipulated animals of FDA-approved drugs that are psychoactive to try and understand if we could come up with an FDA-approved drug that could potentially help this problem. And so what she did is she did her assay, but now she's just trying a bunch of different drugs and trying to find ones that can increase excretion of these fluorescent beads. And so the ones above the blue bar are ones that were able to increase excretion, and the ones below the blue bar are ones that actually made it worse, that they had less excretion compared to the just control. And so what you can see at the top, especially over here, we hit 2 different drugs hit a serotonin norepinephrine agonist, so activating those pathways. These drugs are a little bit nonspecific in that they hit serotonin and norepinephrine. So she went on to do an additional screen on just serotonin drugs. And so the link between gut motility and serotonin has been known for a long time. And actually, a lot of SSRIs, people actually can't tolerate them because they cause so much diarrhea. And so we thought we would hone in on serotonin signaling and try a bunch of different serotonin drugs and see if any of those can increase secretion. And at the dose we tried, one of them was able to do it, which is a escitalopram oxalate, otherwise known as Lexapro, which is a selective serotonin reuptake inhibitor, SSRI. So it increases the amount of available serotonin, and so then those can be received by neurons. And so what I'm showing you here is now she tried to rescue with that. So the top left graph is just on unmanipulated animals. You give them Lexapro, they excrete more, which has been seen in the literature, has been seen in patients. But we thought that one might be, this dose for this drug might be productive. And so on the right, then what you'll see is those fluorescent beads. It's false colored now, so the beads are black. But you'll see how much is excreted in the control DMSO. And then that second row is now treated with that DYRK1A inhibitor. You can see fewer beads per well than compared to the DMSO control. And then with the DYRK1A inhibitor, she then acutely added in this SSRI, Lexapro, for just a few hours. And it was acutely able to move the bowels and produce an increase in the amount of excrement back up to control levels. So exciting. how do we think this is working? And so we wanted to get a little bit more specific than just an SSRI or Lexapro and kind of understand a little bit more about the mechanism. And that's really where her work is heading now. But I want to link it back to those earlier things I told you about cilia and the idea that DYRK1A is really important for cilia. Well, the cilia do a lot of things. One of the things they do is they regulate the migration of those gut neurons into the gut. And so if there are cilia defects, that process is delayed. And so that fits with this idea that DYRK1A is really important for cilia. And so therefore, a reduction in DYRK1A would cause the migration defects of these neurons because they don't have proper cilia. The other thing that cilia are doing, specifically in these gut neurons, is that they're actually regulating the excitability of these gut neurons. And so what she wanted to look at was actually these cilia on these gut neurons, and she was able to find them. And so that little yellow antenna is a primary cilium on a gut neuron in a tadpole, a mature tadpole. And then she was able to use one of our validated antibodies of DYRK1A and again see, just like the skin, now in this neuronal context, DYRK1A is on that cilium of that enteric neuron. And the really intriguing thing here now, thinking about DYRK1A and cilia and gut function, is that they're actually very specific serotonin receptors that are only present on cilia. So for example, serotonin receptor 6 has been shown to be very specific to be on cilia. So those purple lines are cilia in the hippocampus of a mouse, and you can see where there's green is really nicely overlapped with that pink. And so that serotonin receptor is only on those cilia in those neurons. And so the fact that we can see if we increase the amount of serotonin, now we can move the gut, suggests to me that there might be a low level cilia defect in the enteric neurons that affect their excitability, but that increasing serotonin signaling, particularly probably one of these receptors that's at the cilia, can then can compensate for those problems and cause those neurons to still fire. She wanted to do one last experiment to test this hypothesis. And so there are actual serotonin receptor 6 specific agonists. So to activate this receptor that's known to only, at least in the hippocampus, be on the cilium. And so she used now a specific activator of this receptor and asked whether it could rescue. And so now that's what that bottom 3 wells are. The top is what I've already shown you. DMSO is a certain amount of excretion. You inhibit DYRK1A, that goes down. You add back in Lexapro, a general serotonin reuptake inhibitor to activate serotonin signaling. That can get you back at least to baseline. But also the specific agonist of serotonin receptor 6, that can also do it. And so we're excited about this possibility that we could get more and more specific with these drugs to be able to limit off-target effects and start to have FDA-approved drugs that could be potentially helpful for moving the bowels. And so this is our model, and it applies to DYRK1A, the idea that there's this genetic variant, in this case a DYRK1A variant, that causes earlier issues in neural crusts, which will later end up manifesting as craniofacial issues, kidney problems, gut problems, all sorts of things. And then it causes also a problem in the migration, specifically of these enteric neuron progenitors. And then that then leads to GI dysmotility and issues with moving the gut. Okay, good. So with that, definitely want to thank the lab, and particularly, do I have her highlighted? Good. Kate, she has led this work and was very creative in coming up with those bead assays. This is the first time that's been done in Xenopus. And so her preprint is now online on bioRxiv. That's the QR code. And yes, thank you to the DYRK1A community for giving us the idea to study this. And I'd be very interested in your feedback and any questions that you have about this work or its potential going forward. The last thing I want to say that I forgot to make a slide on, but some other work in the lab that we're doing is we're now actually testing patients to understand whether we can have a clinical biomarker for cilia function. right? So told you that DYRK1A is really important for cilia. And so there's a population of cells in the nose where if the cilia are beating, they produce a nitric oxide gas. And so we have a machine that it's just this very superficial tube you put in the nose for 30 seconds, and it will measure the amount of nitric oxide gas. And what we've seen for other genes is that in patients where the gene is important for cilia, you measure the amount of nitric oxide and it's lower than a parent or sibling unaffected individual. And so we're using this now very much as a biomarker to be able to link these genes to cilia function, which can be very helpful to patient populations, because then it also says that child should be screened for respiratory problems, kidney function, as well as other potential organ systems that could be affected that may not come to the surface immediately in a clinical setting. And so if people are interested in coming to the Bay Area, we also travel around a little bit with this machine. Please reach out to me. I can send a flyer to the DYRK1A group so that you can send it out. But it's a 30-second non-invasive test. We do it in the lab, or we can find a place to meet you. And it's IRB approved, of course. And we usually either do a sibling or a parent to control for genetic background. So thanks for your attention. Happy to answer any questions for anybody that's in the room. Thank you.
Thank you so much. Questions? Hi. First of all, we live in the Bay Area. Yeah.
Come on. So just email me. I'll hook you up with the clinical coordinator, and he can find a time to bring you in 30 seconds. It would be great to get that kind of data on DYRK1A. We have it for other genes, and it's been positive.
That would be great for us.
I'd love to meet you. Come on by.
Hi. Jordan does have a lot of constipation issues. I guess my question would be, since we've been dealing with the constipation issues for a while, we've tried multiple different medications, and it seems that it hasn't really been working well. Is it kind of a lost cause to try these medications like Senna and Miralax and all that?
So I'm going to give you a big caveat that I'm not a physician. I don't understand anything about medications. I know in talking to these rare variant populations that they've tried a lot of these things, Miralax and fiber and things like that, and they don't seem to be super effective. But that's why we wanted to go for something that's FDA approved, because the idea is that maybe we could go quicker into folks. But I'm not a physician. I'm not going to tell you you should do this. I think it would be worth the foundation thinking about where they want to go with this. We are starting some rat work with this. And so we don't have a DYRK1A rat yet, but we could think about getting it and then doing it in the rat more as like a preclinical model. And in that case, then the rats will be heterozygous variants. Whereas the frog, we're doing a pretty strong loss of function. And so the rat genetics and the rat GI system might give us more information about whether, acute Lexapro could actually move the bowels. But certainly something to talk to physicians about. I am not qualified.
Yeah, thank you.
Thank you.
Another question in the room? Also questions online for those of you who are still monitoring the Q&A chat.
Thanks so much, Helen, for the wonderful update and talk. It was very informative, and it's fascinating part, the porta potty assay for the tadpole. It's just like very, very intriguing, very intriguing. And, we talked a lot about sort of the neuron migration. Do you have any more sort of data on just the quality of the neurons themselves? Because, in a lot of other papers and just sort of background, I mean, we've seen the impact of, or, observed impact of DYRK1A just dendrites or axons. And so do you see a lot of those same impacts in a tadpole, or is that something that you can measure? And then, I'll just start there and I can ask a follow-up.
Yeah. So I'm anticipating that you are going to have the same question as reviewer number one, maybe. So her paper is in review right now. What we're doing while we're waiting for revisions is she's doing exactly that. She's trying to look now at mature tadpoles, look at the morphology of the neurons, look at the proportion of different neuronal subtypes. So in a colonization of the gut, the serotonergic, the serotonin neurons actually colonize first. And then they actually direct the boat for a lot of the later populations of neurons that come in. And so you could imagine that problems in that early colonization population could then trigger even further disruptions in the proportions of neurons that end up there. So yeah, she's looking at morphology of the neurons once they get there, the proportion of different neuronal subpopulations once they get there. And then she's also using that fluorescent reporter of neuronal activity, the GCAMP, to actually see, are those neurons firing or are they just not? And then she can add in the Lexapro and see, now do they fire? Is that really how Lexapro is working, or is Lexapro working in the central nervous system and then there's feedback, or there's a bunch of different ways Lexapro could be doing its jam. But that's certainly things that she's doing now, but we don't have data on yet.
Great, thanks for sharing that. The other question I had was just related to, I guess, the more global picture. If we're talking about the association between autism spectrum genes and sort of the phenotypic gut issues that we're seeing, and from your work, you've kind of pinpointed it to Zircon-A impacts on the cilia. So is that what's also being seen in other autism spectrum genes? Do they impact the cilia, or do they impact the neural crest cells in different ways that manifest similarly in gut issues?
Great question. So Zircon-A has definitely been our pioneering gene, and what we keep seeing is what we see with Zircon-A usually translates to all these other genes. And so we have a paper now that we will be submitting, at least on bioRxiv, in the next month or so, actually now showing that most of these other autism genes, even ones that have synaptic functions, even ones that have chromatin functions, they are also at cilia and really important for ciliary biology. So we think that there's a lot of overlap of mechanism, even with genes that y'all may not directly associate with Zircon-A. So things like SYNGAP, things like STXBP1, things like CHD2. You may know some of these patient foundation groups, and I think you all have a lot more in common than it is currently thought of.
Yeah, thank you. And my last question is just related to kind of what was mentioned earlier. A lot of our kids, they're you go to the GI doctor saying, hey, try Miralax, or try fiber, these sort of things. I'm not sure if there's like an exact way you can do that in a tadpole, but is there a way that you can give a tadpole Miralax, or you show that, hey, it's specific to these serotonin receptor 6, using Lexapro is really the only thing that's working. We've given this tadpole Miralax, or, what's the other one I'm thinking of?
Metamucil. Yeah.
Metamucil, right. I should know that. But yeah, like showing that even with the addition of like kind of what's generally recommended, and clinically, like you still don't see increased gut motility or excretion.
I think that's a great idea. We haven't done it. We were really going after psychoactive drugs that are FDA approved. But I think it would be a really nice foil to see, Lexapro specific, or it may not be. One of the other things Kate is doing now is she had a few other hits that were like acetylcholine and a few other neuronal populations. So she's trying to see if those can also rescue, or if it really is specific to serotonin. But we can add those in. That's the beauty of the tadpole. This is a 1-week experiment that costs me, I don't know, Kate's salary for the week. So like we can do that quickly. And I think it's a great idea to do, to establish, this isn't just your typical constipation. this is like its own thing, like Hirschsprung's is, right? Like those kids have to have bowel resections, and some of the conversations I've had, some of the issues are that severe as well in this population, right? So I think really emphasizing to GI doctors that this is more like Hirschsprung's than it is like a typical constipation. I think could be really helpful for them. And my hope is that this publication can help you all start those conversations with clinical folks and think about whether this could be helpful. But yeah, if you have anything you want me to put on those tadpoles, I'll put on those tadpoles. No problem.
Any other questions in the room? I don't know how many other parents were, their children were tested for Hirschsprung's. I thought it was, and they didn't find anything. So you say it acts like that, but obviously it's a little bit different.
Yes, absolutely. So sorry if what I said made it sound like those could be equivalent, right? So in Hirschsprung's, the neurons actually never even get there. So then there's a huge, basically, die-off of the gut tissue, and that's what ends up being lethal if you don't resect back the gut. I think with Zircon-A is going to be a much more subtle issue where the neurons probably get there, so it's not lethal. But then there will be differences in the amount of activity of those neurons, such that it's basically a very, very, very mild version of Hirschsprung's. But I agree with you. By no means am I trying to say that you could do sort of the Hirschsprung's tests or things like that and have them be equivalent. But I think for GI doctors, sometimes they don't understand the severity that can be going on with rare variant populations compared to a typical population.
I'm not sure about other parents as well about the pain, because our children don't feel the pain of having to go to the bathroom. So the size of their stools are so large.
Yep.
And I don't know if it's because they just can't feel it anymore, or they never. My son never complains.
So that's very interesting to me biologically. And so would you say that high pain tolerance is also translatable, for example, to spicy foods like jalapenos or to heat or things like that?
I don't know the spice food, but for my daughter is high tolerance for any external pain. Like she got her feet really bad once, blood everywhere, and she didn't even blink.
So that's very common for a lot of the patient organizations that I work with. The one that's coming to my mind is SYNGAP1. Those kids have an incredibly high pain tolerance. And actually, a lot of the sensory cues that relay heat and pain are through cilia. And so a lot of the receptors that need to be activated in order to sense pain are on cilia. So a high pain threshold is certainly consistent with the idea that there could be a ciliary mechanism at play here. But again, there are many genes where I think that is a similar finding that are also on cilia. And I've also heard from these other patient groups where the stools are incredibly large and they're having to do enemas every other night. And yeah.
Yeah, so, and just to follow up on that, just interested to know if that's something that you could model in the frogs, or if you see that in frogs, because I do think a lot of the parents, it's somewhere, it's like, it's somehow just, it's so slow that it just builds up, you know, and it's not even really the, you know, just slowness of it, but that it's kind of like backing up. And I didn't know if that's like something phenotypically, or you could also see in the tadpole or frog.
Yeah, so that's on Kate's to-do list for later this summer, because we have those fluorescent beads and we can see them in the gut. That is something we want to look at. Is it just a buildup, and is it spatially? Is it building up in one particular part of the gut, or is it distributed, or things like that? So we could look morphologically now that we have these beads that we can see to see what happens there. So hopefully we'll have some idea, at least in the tadpole, what that looks like. But again, my hope is that we expand this out to the rats. We're doing the rats right now for SYNGAP and for SCN2A. And so if we can get anything good there, then I can try and get in DIRC1A as well.
Well, this might be a good time to also just plug in, you know, we do have, like an RFP, you know, for grants for, you know, modeling DIRC1A, especially, you know, a lot of the work that you're doing that is applicable, you know, for gut motility and constipation. So, you know,
I should apply for that. I've been
Yeah,
funding
yeah.
it. I've been funding all this myself, but great. I didn't even know
Yeah,
you
so
had.
there's
I didn't
definitely,
even know you had grants. So I'll
yeah,
look into that.
than that, and, you know, I'm sure the community and the group would be open to your nitric oxide, um, uh, you know, study, and, you know, we can just get, you know, more information and pass that along with the community and figure out a way how we can, you know, help participate in that research.
Yeah,
But-
absolutely. And if you ever have family meetups and stuff, I'm sorry I wasn't able to get everything in line for this year, but whenever you have those, if you can send them to me, I can send one of my folks out there to do testing there, so that if you have a concentration of folks together anywhere, I can send somebody and add that to the IRB. So just keep in touch. But yeah, if you're in the Bay